I'm going to talk a bit about the sort of work that we've been doing here at the lab. This is essentially part of this ERC grant that Sebastian has called Grow AI, which is about studying growth and development and self-organization in the context of how it can help AI. But also, I think also just trying to take some of the ideas and see what we can learn about biological systems, right? And about the computations involved in self-organization and whatnot. So what I've been working on lately is this idea of using morphogens as scaffolds for organizing systems. So I want to first acknowledge the people that have helped me mostly with this work. Elias, who is my main co-author, and then also Eleni and Sebastian, which have been very helpful. Okay. So maybe a bit of motivation, and I don't have a biology background. My background is computer science and cognitive science. So I may say things here that are I'm going to try not to say too much about biology, but if I say something wrong, please feel free to interrupt me and correct me. In fact, feel free to interrupt me whenever you want and ask questions. I'm happy with that, and I actually prefer that than just talking for 40 minutes. So yeah. Okay. So usually, there's a lot of research that is mainly involved on how self-organization can serve developmental systems, right? And a lot of this work is focused on or computational work is focused on how this process can unfold from the point of view of having distributed systems and collective intelligence. You have different components and they have to communicate and self-organize and reach some final state, some final developmental state. One I guess model that has maybe become popular in the last few years are NCAs, and I don't think I need to explain much about NCAs to this group. The NCAs are cool in the sense that they're a simple model, at least simple by today's standards, that allows us to probe and see how different self-organizing systems can behave, right? So what I guess what NCAs, you can train them and they get really nice patterns and whatnot, but maybe a more interesting question is, well, what can we say about self-organizing systems if we have and development if we have an NCA? Well, for starters, they're really hard to train, or the standard NCA maybe not so hard, but as soon as you try to do maybe more interesting things like invariant, like isotropic NCAs and invariant NCAs, they become really unstable. They're not really easy to train. Scalability is also an issue. Trying to extend the number of developmental steps or the size of the substrate can really hinder training for NCAs or how well they can achieve their target. But they're not very scalable, which is a bit disappointing because, as we can probably see, biological development is very scalable. So there are things missing from NCAs. And one main thing that NCAs need to do in order to start this process is they need to break symmetry. You have a bunch of units and they're all identical, and in some way you need to decide which unit is going to become what, so that they can take on different roles within this collective system, right? So this is the start. This first first idea of needing to break symmetry is what started like our motivation to get into this idea of morphogenetic gradients. So first of all, biological development seems to not be just about self-organization, at least in my view. There's many different ways in which, or many different mechanisms that biological development uses to signal to cells in a developmental substrate what they need to do and how they need to self-organize. And one of them is this idea of morphogenetic gradients. So why are morphogenetic gradients? Well, I guess for me they're interesting because, as you can, I don't have a pointer. I don't have a pointer. Well, I'm going to continue like this because this is easy for me. Biological development Slide the navigator. Slides only. So the reason why morphogenetic gradients are interesting is because you can have this sort of this chemical gradient that will cover some developmental substrate and give positional cues to the cells within that substrate and allow them to use that signal to better guide self-organization. You don't need to now propagate signals across the whole gradient in order to decide what to do. You can just use that positional cue to help you, and you only need to propagate information maybe locally. So essentially, this is, I guess, one of the benefits that you can see with it. The established axis of development, guide the determination of cell fate, separate the where from the what. And one analogy that I like a lot is that it's an analogy to maybe higher level cognitive systems. Is that it's a bit like variable binding. You can establish that at this position in space, you want to build a particular structure. And for a different species, or they're different, even across the same different organisms from the same species, that can vary, and you can still get, you can reach your final target appropriately. This is, why is this doing this? Okay. So the idea is that we're going to take this idea of morphogen gradients and we're going to combine them with NCA somehow. So how can we do it? Well, one simple way of doing it is that we can define a function over coordinates in the developmental substrate. And this is fairly easy. You just take your positions, x, y, within this 2D substrate, and you define a function, and you give, and that function gives you a value, which can be a vector value. You don't need to just do scalars. And we can use smooth functions, and this is important to some degree. And these are like examples of smooth functions that we're going to use. For example, like a Gaussian function in the top left, or you can use periodic functions, or you can use directional gradients that give you, that slowly degrade from each of the boundaries. And you can use that to essentially establish in which position of the field the cell is located, and thus what role it needs to take on. And we can use them to condition NCAs locally. So the way we're going to do it is you take your standard NCA and you you do your update or your perception. So you apply the kernel around your cells, and that propagates information across the L. And before we perform the update, we take the morphogen fields that we have defined, concatenate that to that perception vector, and pass it to the update function, which is a standard, like it's the same standard MLP that was used in the original NCA work. Once we've done this update, we proceed as usual, update, and then repeat this process for several steps, and until we converge to some value, or we reach the final number of steps. Okay, so we have this system now, and this is the example of the kind of data that we're going to use. So these are like example morphogen fields on the right, and some target emojis that we're going to use to test the system. Importantly, I think it's worth mentioning, these emojis are all symmetrical, and they have different types of symmetries, but they are symmetrical. And that just helps with training the system. Okay, so this is some reason not working. Okay, that's working. Okay, so we're going to test.
Sorry, do you mind if I jump in with a quick question here?
Sure, yeah.
So in your previous slides, you looked at 2D mappings from a morphogen to a function, and
like here.
Exactly. But most of these morphogen gradient pictures look like they're kind of 1D-ish.
They're 1D-ish?
I mean, I guess maybe not, but I guess maybe what I'm saying is that there's just one morphogen and not two morphogens.
Well, but we can.
It's a 2D picture, but using one variable as opposed to two variables. Is that the right understanding of what you're using?
I guess what I meant when I said 2D is that you give a different value for each x, y. Now, for some morphogens, sure, it could be the case that the same x has the same value, or sorry, different y's or different x's have the same value, because the morphogen is only varying in one direction. It's not varying along x, but it is varying along y. Like, for example, in the case of the...
But it is just one morphogen.
Well, we actually, we can, and we have several, I think I'll get into this a bit more later, but for example, we can concatenate more than one morphogen, right? We can give the system more than one morphogenetic cue to guide it. We don't need to just give it one. We can give it several different channels, right? We can use the last 5 channels of the hidden state and say that those 5 are morphogens, and they can all be different morphogens, like in the example below. And that, it's valid, right? Is that clear, or am I still not?
Yeah, totally makes sense. Thanks.
Can I consider this concatenated fields as similar thing as position encoding in transformer?
Yes, that is a that is a good analogy. Yes.
Right. Interesting. Thank you.
Yeah, no worries. And this is a bit what I was referring to with separation of the where and the what, right? You can use this to signal, this is your variable, which is, I don't know, top left, or it's all spatially located or organized variables. And then within each one, that should help you say, well, what I need to do here is an arm or a leg or whatever, right? But that's essentially the idea. Okay, so we have, I think one thing else worth mentioning here is that I'm going to mostly work with invariant NCAs. So the perception kernel is invariant to direction. I'm not using the standard Sobel ones. I'm just using a Laplace perception. So it has no idea about direction unless I say otherwise, which is the case of, for example, the second example, right? So we have like a baseline here where we have all these, the blue dots are cells, and they are enclosed in this substrate. And we have, can you see my cursor, by the way? I'm not sure if you can see it. Okay. So this baseline just uses boundary conditions. And the idea here is that you use boundary conditions, but there's only a distinction between outside and inside, right? So inside, everything starts at 0, and the outside, it's always -1, for example. In the second example, this is a standard NCA. It uses Sobel filters, which I've sort of tried to illustrate here as some sort of polarization of all the cells. They all have a sense of direction, but not only do they have a sense of direction, it's a global shared sense of direction. They all know what the canonical north and canonical east is, and so on and so forth. Then there is a second example here where we are going to use, we're going to give each boundary a different value to help maybe break symmetry better. And finally, there's this final one here. Apologies for the not so good figures, but here you're using mixed morphogens. So we're using the morphogens that we saw before, like all 5 of them within the substrate. And so what's interesting here is that if you now try to train each one of these to reproduce a particular shape, in this case, a beetle, the isotropic NCA cannot essentially break symmetry correctly. So it will try to break symmetry using the boundary conditions, but because all the boundaries are identical, it cannot fully reconstruct the image. It just produces symmetrical patterns that it cannot reconstruct. If we try with now the Sobel filter, it can do it. It is, it can do it, but you'd have to use this sort of maybe unrealistic, or at least in my view, unrealistic notion of this global thing where everything is polarized, and I'm not sure this happens most of the time. It's also very unflexible anyway, because you have to establish this, and this is canonical, and if you change it, or even give each cell their own direction, it breaks. If we now use different boundaries, different values in the boundary with an isotropic NCA, so now it has no global sense of direction, you can reconstruct, and that works well. Though it is maybe not the greatest reconstruction, it also is a bit slow. It takes a bit to converge. If we now try with the other one, you can see that the model doesn't need to break boundaries, as you would expect. It just uses the positional cues to figure out what it needs to do. The positional cues are not maybe enough to tell each cell what it needs to exactly be. It needs to still coordinate a bit with the cells around it, but it can converges very quickly, you know. And we can actually
Milton, can I ask a brief question?
Sure.
Are the NCAs you're using here synchronous, or do you use asynchronous updates for the cells? Because this could also lead to symmetry breaking due to time delay or something.
Yes, and in here I'm using synchronous, mostly because it's just easier to train. We did try with asynchronous. The problem is that because the asynchronicity is sampled, like if you try it, for example, with the isotropic NCA here with only one uniform boundary condition, it doesn't make a difference, right? Because for each run, the stochastic updates will just be different for each cell. So the cell has no reliable signal to use to break symmetry correctly. So it doesn't make a difference, right?
What you could try, what would be interesting, is whether the cells could come up with a direction that is important to them when you rotate the solution and you allow it to grow either upwards or sideways or the other transformations.
What do you mean?
If you don't train for one particular emoji that only shows into upward direction, but you also allow for the different targets that go sideways or all the transformations that are necessary here or allowed, like rotated counterclockwise by 90 degrees, then the cells could spontaneously during development form a direction of orientation towards left, right, or something.
That could be. I'm skeptical that would work, but you know, it's an empirical question, so we can definitely try. Okay.
I have a continuation to that. What will happen? You stop on step 40. What will happen on step 41 or 50? Is the pattern stabilized or not?
No, the pattern doesn't stabilize, and I think this is just a common thing with NCAs. I think this is maybe one of the reasons why the original NCA used this idea of this pool, right, where you kept track of trajectories and resampled them and retrained from them to like sort of force it into converging towards this towards the target and not move and be stable. We haven't really tried this here, like to maybe be too. I would say we haven't been too worried about whether it will stabilize at a pattern. Although I think that is, I agree that that's an important question. Currently, it doesn't. I don't know if I don't like this idea of this pool that you need to resample. I don't think that's a weird trick to make it work, but not maybe what's happening. Probably not what's happening. It's definitely not what's happening in biology. The morphogens don't seem to help much in this regard. So maybe another mechanism is better. I think maybe one thing that is maybe impeding. I think a better way to solve the issue of robustness is having some way to encode your target that basically says how close are you to it, and just forces the update rule to converge back. You know, not so much by keeping track of what state you are, but how far away you are from your goal, right?
Right, but that goal is a global goal, so all the cells get a signal from everyone. Like I don't know, a bird eye view that see the whole system and say, oh, you need to go move, you need to move right, move left.
Yes,
Well,
yes, but the goal would need to not only be, so the goal needs to be encoded in somewhere globally. I guess in real system would be DNA or something. Who knows? But it also needs to be able to, I guess, not only know what the target goal is, but am I close to my role in the target goal, right? Because it doesn't make sense for you might be missing a leg, for example. It doesn't make sense for the cell that's already built a hand to say, oh, he's missing a leg, I need to do something. No, you're fine. You don't need to do anything. You're already a hand, and that you need to be. The error is somewhere else, right?
So there is a error signal that say, oh, you reach to the maximum stop. That's what you're saying.
Yes, something that says, you've reached your local goal, your particular goal you've reached, you don't need to move. And obviously there must be some way in which all those local goals coalesce into the target, the target morphology. Is it a human? Is it a bat or whatever? But I do think it needs to be more local than that. And I don't know. I mean, I guess the morphogen fields could help with that, but I think it has more to do right now, or in a real system, it has to do more with how we're using the NCA and how we're using the update rule than whether we have morphogen fields or not.
Thank you.
Yeah, I feel this is exactly what diffusion model is doing. It's estimating the added noise and do the denoise. Actually, we previously had a paper train NCA via diffusion model, and the trick is the diffusion model is not using like UNet, which perceives the entire picture, but it's a design in purpose that only perceive the local patterns, and you can really get NCA from that, and it will stable at the pattern you train on.
Okay, that makes sense, because we had this similar idea maybe a year ago and never did anything about it. But I could totally, if you have like a preprint or something, I would definitely be interested in seeing that. But I totally buy it because that is indeed how diffusion models work, and I could totally see an NCA being able to do that.
Yeah, I shared in the chat. If you're interested, we can talk further about that.
Okay. I'll check it later.
Sorry, can I ask a question before we go to the next slide? So the last example on the right, the morphogen. So it is again a symmetrical NCA. All nodes have the same state. The boundaries are all the same, and the only thing that breaks the initial symmetry is the morphogen field that goes from left to right. Correct?
Not only that, yes. Not only that, there's actually no boundary here. Now that I recall, in this particular example, it's a torus. So it's the convolution is wrapped around the boundaries. There is no boundary condition for it to break. Right? It needs to use the morphogen cues to self-organize. There's no way to know if you're at the edge or not.
Okay. So I guess then.
Yeah, go ahead.
So the only thing that breaks the initial symmetry is the morphogen field, right? And it's just a one-dimensional gradient that goes from left to right, right?
No, I mean, well, it's it is the ones down here. So for that example, I just went a bit overboard, and the ones at the bottom here. Wait. Sometimes my cursor disappears, other times it appears, and I don't know what the logic to it is. But the ones that said morphogen fields at the bottom. So we just give it a mix of morphogen fields, directionals and Gaussians too. So the Gaussian helps you because it tells you how far you are from the center, and then the directional ones give you cues as to what position you are within the substrate, XY positions. So you could argue that it's a strong signal, but yeah.
No, I thought it was a weak signal because what I see on the slide is just a one-dimensional gradient, but the beetle itself changes in the top-down direction, not the left-right direction. So I was wondering how you were able to train your system to do this with a one-dimensional gradient. But what I'm hearing is maybe you have a much stronger, more complex gradient.
I mean, you can train the beetle. This is an example where it's a bit drastic. You can train the beetle with only two directional morphogens, and it works. Just give it XY position and then train it, and it works. The more morphogens you give it, especially if they're not redundant, obviously, the more information it can exploit, and the faster it will converge, right? But
I guess it has to be at least two directions, right? Not just one.
Yeah. And we also, if we give it more morphogens, we also give it less hidden channels. So there is a normalization here of the number of hidden channels that the state can use. So if you have no morphogens, you get the maximum number of hidden channels, which is 12. And if you have, so that's hidden channels plus then the RGBs, A. So you get in total 16, right? If you have two morphogens, then you get 10 hidden channels. And in the case of this one here, it has 4 morphogens, so it gets only 8 hidden channels.
Okay.
Okay, so we can actually sort of systematize this a bit. So we can run and see how fast how well they converge to a target, and also how fast they do it. So symmetry breaking is really, I think is really helped by having these sort of scaffolds. And you can see that the one that uses the mix of several morphogens, and even though it's on a torus, it can converge to a really good, it can be really faithful to the target, but also it can converge pretty quickly. So this is, I guess, just the point is, it's helpful to have these morphogens because they help the systems figure out where they are, and then there's less need to propagate signals across the whole field. And so you just get more robust and more and faster self-organization, which I imagine is beneficial in real biological systems. Okay, something happened here, and this is not doing the things right, but anyway, we're going to test now this idea of generalization. So the idea of generalization is, if you have a target and you train it with one particular morphogen, can we now change that morphogen in some way, manipulate it, and can you get some form of generalization? Can you one-shot generate a novel pattern? So yes, we can do this under some conditions. And the conditions are essentially that you need to respect the symmetries within your target pattern in order to get this idea of generalization. And I guess once you see the examples, it may be not that surprising. So I'm going to illustrate two examples. One is rotation. The other one is removal of parts. Addition of parts also works, but I couldn't find the simulation where I'd done it. So we're going to do removal only. But the idea is you have a target here, like an 8-petal flower. And if you can train it with either a morphogen that is aligned, like the one here. So you can see that the arms are aligned with the final petal. They don't encode everything about the petal. The developmental system still needs to say, well, this is the area where the petal is going. I still need to build it, the width, and the width is not the same at all positions of the let's say arm. And there's different colors and whatnot. So it's just that. And in this case, we're just going to use this morphogen. There's no other morphogen in the system. You can also misalign them. So you can take the morphogen, rotate it as on the right, and now they're misaligned. You can still train the system to do so, to reconstruct this same target, which is also computationally more demanding. The NCA now needs to do more work because the cues that it's getting are a bit misleading. So it needs to sort of figure out that it needs to propagate some signal to, in clockwise or counterclockwise, whichever one, to the final position, so that you could build the structure there.
Milton, is this because the cells are not alive at the signals? Like the signals and the alive cells are misaligned here?
Is this because the cells are alive?
Because
yes.
I mean,
yeah, that I mean, but even so, this I'm going to show an example with a system that grows, but it's the same with the system doesn't grow. You still need, let's say, all your cells start alive, but with a 0 hidden vector. They still need to. They're getting a cue that says, "Oh, you're at the arm," but your target is shifted by some phase or some amount of radians or degrees. You still need to propagate that information. Well, the only difference is maybe that the propagation can happen at once, right? You can do the propagation of all of them at the same time, while here you need to build and maybe do something, but the issue is the same, right? You're getting a cue that you're in a particular position in space, which is not the one aligned with your final target. Okay, so let's see, if you train it, you take the morphogen and you train it, and the result is this. Now, if after training, you've trained it with this thing that is aligned, and you are getting the final results, and all is good. If you train it with this one, what is the result? Well, the result is, the result is that you can get the rotated pattern. It's not that great. We need to rerun this simulation with like a better resolution and whatnot, but it sort of works in this sense that you can see that the structures are emerging and they are rotating the figure in the correct way. If you do this without it, and I apologize, I had a slide with this, but I couldn't find the right simulation that was doing it, and something was not working. I probably changed some code somewhere. But at one point we had an example that we showed at A-Life where if you do this without with misaligning the gradients, it fails. And we can do this systematically. We can test this systematically, which is what we did. So on the left, you can see what happens if we train the model aligned with the morphogen, and then we slowly rotate the morphogen field across all possible angles. And you can see that it correctly and test that it's reconstructing the rotated image. Is it correctly reconstructing the rotated image? And it does. It can it perfectly reconstructs all the rotated angles of all the rotations of the target. On the other hand, if you train with a misaligned one, first of all, it's harder to train, so it doesn't reach the same level of precision of the final target. But I think more importantly, if you rotate the morphogen field at test time, it struggles to reconstruct the rotated version of the target. And you can see there on the right, sorry, that as you move away, you change the degrees that you've rotated the morphogen, and thus the target that you expect, your mean squared error just grows quite a bit. Questions? All right. So I think rotation is nice because it's very visual. It helps. I mean, they're all very visual, but it helps, I think, illustrate the point in a very fun way. But now let's do a different one where you have to remove petals. Okay, so let's say you train your system with this morphogen to reconstruct this as before. And now say you train with a non-matching morphogen. So what is a non-matching morphogen in this case? One where you remove the arms of your morphogen field, right? So the system needs to start from this and still needs to reconstruct this eight petal flower. Now at test time, you change the number of petals in the morphogen field. You expect that you can now reconstruct, or the system will produce a flower with 7 petals. So what happens? Well, I'm going to plot on the x-axis the number of arms in the morphogen gradient during evaluation, and on the y-axis, like each row, the number of arms in the morphogen gradient during training. I'm going to try to guide you through this because I don't know why I keep. Oh, here it is. Sorry. Okay. So here, these are all the ones that you used during training. So each of these models was trained with this particular morphogen value, and these ones here were the ones that were used during testing. On the diagonal, you should see perfect reconstructions of all the flowers. Why? Because these are the ones that they were trained, and they were tested with this same one, and they just produced the same target. Now, when you vary the morphogen here at test time, you imagine you would see here different number of petals in the final target. But that only happens for the model here at the bottom, the final row, which was trained with an aligned morphogen pattern. So as you take this model and you vary the number of arms in the morphogen field, you see that it manages to successfully reduce the number of petals in the flowers. Now, it also happens for other systems. For example, here and here, they manage to correctly reduce the number of petals in their target, mostly because maybe the symmetries match in some way, right? If you have 8, or if you have you manage to match them in some way, you manage to overcome it, and you can still do it. But in general, they fail to produce the correct target when you manipulate. So we can essentially the point is we can get 0-shot generalization to novel morphologies by manipulating these gradients, at least for this case. And I imagine there's ways to construct them so that we can still do it for other cases. And we can systematize this again. If you plot the error that you get as you vary the number of arms at evaluation for the different trained one, you can see that error just goes up for most of them, and then it just dips a bit at the end here. While for the base, the one that was trained on the matching one, here the error just is very low throughout.
Can I quickly ask a question?
Sure.
Yeah, I think this is very cool. To me, it looks like the morphogen gradients. It looks like the end result is basically a set intersection of the trained morphogen and the evaluated morphogen, if you really think about it. Is that a good way to think about it?
A set intersection.
Yeah, for example, if you take the last row, right? So the trained is what is it? 1, 3.
It's this one here. It was
8.
trained on this one.
It's 8 arm, right? And so if you show the plus, then the intersection of the plus and the 8 arm is just a plus.
Yeah.
So is this kind of, I think it kind of holds for all of this, right? So if you just compute a set intersection of the trained morphogen field and the evaluation test field, you can kind of guess what the outcome would be.
Not exactly.
Is that...
So not almost, but not exactly. So what we did to get from here to here was not just remove these. We removed them and also made them still equidistant in angle, right? So the angle between these two here and these two here and all of these is the same, right? And so is this the case here, right? So there's always a bit of a displacement between these things and these. They're not exactly matching, and it's not just a set intersection. If you do a set intersection between the images, you will cut the petals at some point. So it will not be like the full petal. You will just cut it because they're not really overlapping. See what I mean?
Yeah, I get, yeah.
I mean, maybe it's not clear here because maybe the images are too small, but I did make sure that it was not a set intersection, mostly because of that. Because then it's maybe not. I mean, I don't. We can discuss whether, or we can debate whether how trivial or complicated this is. But I wanted to make it as hard as possible, with the same idea of just varying the number of things.
When you use the morphogen, how many bits of precision are you allowing the NCA to read? Full float-in point?
Float 32.
Right. So I wonder if, I mean, in biology, of course, you cannot read a signal with 32 bits of precision. If you're getting 2 or 3 bits of signal from a morphogen gradient, you're doing well. Any more than 2 or 3 bits is not really biologically realistic.
Okay.
And I wonder if that's kind of, you're giving the NCA the ability to grab so much precision that it's, I mean, in principle, with a 2 morphogen vector field that, varied from 0 to full, one varying from 0 to full in the X and another varying from 0 to full in the Y, you of course could fully determine your position and there would be almost no training needed. And if the more, I wonder if the more you allow the NCA to have access to this unrealistically precise cube, the more it will wind up depending on it. And the more, when you change the morphogen field, as you've shown here, the more, since it depended unrealistically much on the morphogen field in the first place, the more it's going to be tricked when you change it.
That could be. I agree. Yeah, I think it's interesting that you mentioned the other. I hadn't thought about the position that we give the systems. I don't know, because I guess there's 2 questions here. One is, how relevant is this for biology, or how well does it match? And the other one is whether the comparison we've done is fair. I do think the comparison. I take the point about similar or faithfulness to biology. I think the comparison, though, between the systems as they stand here is still fair, because they all have this precision that they can use, right? I think the issue here is that if the morphogens are aligned, the system says, well, I know that if I see this Q here, it means I need to build a petal. And while the other one needs to do something more complicated that is just not inducive to generalization, right? And so the one that just has the morphogen line has a much easier job, right? Which is the whole point. Is the point that this alignment of symmetries just helps things to move at a better pace and to generalize in a more systematic way. So it maybe gives an argument for why it's good to have morphogen field or scaffolds that are aligned strongly with what you want to get, and not just give, because you could just give positional information, right? You could just have a gradient that goes from the grades from top to bottom and the grades from right to left to right, and that already fully determines something, but you would not get this behavior, right? So it's not just about giving positional information, it's about giving instructions of what to build where, right? And I think that is, I guess, the point that we're trying to make here. Does that make sense?
It does. And there are some biological systems, I think the classic one that people, have experimented a lot with is mouse fingers, where there is a morphogen gradient that's been shown experimentally to have like, a reaction diffusion pattern with 5 peaks. And each of those peaks during embryonic development winds up corresponding to a toe. Occasionally, because these patterns, these morphogen patterns have an intrinsic wavelength, and there's always some give and take over there. Occasionally, the morphogen field has 6 peaks instead of 5 peaks. And indeed, when they look at that, the mouse has 6 toes. It has faithfully followed its morphogen instructions. But I don't think that's a characteristic of all biological systems.
No, I agree. I imagine that it's not a characteristic of all biological systems. I guess here the point is systems who do follow that might have a benefit. They're more evolvable. They have more types of morphologies that are easily accessible to them. Right? And in this case, they're the one that was trained with the aligned morphogen, can access all these different morphologies just by varying the morphogen field, while these ones here maybe have access to the target one here that they reconstructed and one other, right? But they cannot, here reconstruct. This is an image, and an image is not really a morphology. It's just I guess, a toy example of what you can do. But the point is that generalization is a stand-in for evolvability. How many things do you have access to if you make this symmetry match? And it seems like this is just more generalizable, which in practice means it just can evolve to different things more easily. While the other one would need the NCA parameters itself to evolve. Does that make sense?
It does. It's a pretty cool property, the ability to have, substantial changes in form from small changes in morphogens.
Yeah. Wait, I have 10 minutes. I actually have quite a bit. Okay. Okay. So maybe we'll skip this a bit, but I'm not sure rotation. No, it's a good illustration, but I think rotation is a bit weird in terms of biological development, because what does it mean to rotate a flower? I guess you can rotate structures within your system, but not clear to me what that means in practice. I do think the petal idea does suffer a bit from this idea of like reverse causality, which I guess maybe in this group is not or top-down causality, which I guess in this group is not an issue, but maybe in the broader biological literature might be assumed like, well, why would this try to converge to a flower that has 7 petals? When it's not specified beforehand. The morphogen specifies what, and then from that you keep going, and if it doesn't specify, there's no point reaching this particular target. So I'm a bit fuzzy on the implications beyond this simple generalization. I'm much fuzzier. I'm fuzzier on the idea of how to properly speak maybe about this in a biological context. Okay. I guess to like wrap up, I don't know if how much time do we have past 4? Am I past 5 my time? So I'll try to maybe do this fairly quickly. the last thing is that for now, we just use these morphogen pre-patterns like as fixed functions. And we've used them to manipulate and show that they have benefits for symmetry breaking and whatnot. But what if you're trying to learn them? And we can learn them because we can always take this function f and give it some parameters. And then we can train the whole system end to end. So what does this function now do, right? You still take the coordinates, but now you have an MLP. And the MLP produces some output. And importantly, the MLP has sinusoidal activation functions within the, like here, in the hidden layers. And this is a model in that in the machine learning, especially in the vision computer graphics literature, is called it's an implicit representation, and specifically this one is a siren. So implicit just means that it takes positions x, y, and outputs some vector for each position. And siren is just because it uses sinusoidal activation functions. So we can take that. We can then take the output of that and populate the visible channels of the NCA. And this is an example of a pattern that you can generate with it. Then we can run the NCA parameters, and we can run this for several steps, and lo and behold, you get your typical salamander at the end. Now, this is a bit overkill for one parameter, for one pattern, but we can actually just use an embedding to condition it across a data set. And the idea of the embedding is that you modulate the activation. So you don't affect the parameters of the dense layers or the NCA. You just modulate the activation values after each of these layers. So for each element in your data set, you get an embedding, and that embedding helps you scale up or down the activations in this the output activations of each of these layers, and that already allows you to control these systems. So just to give a bit of illustration, if you train just keep messing this up. If you just train this here to directly output your target, and with no NCA development afterwards, then you get sort of these patterns. So this is a very shallow one. You can go even shallower, produce even more coarse patterns, but you can, because I wanted to show that you can get something that's close to your targets. If you train it with a depth of 5, you already get pretty good reconstructions. Now, if you take the one at the top and combine it with an NCA, this is sort of what you get. So the shallow one learns coarse patterns, the deeper ones reconstruct the data set. And by the way, there's no growth here, it's just self-organization, and we'll see why in a bit. But I think what's interesting here is that you get some form of, for example, like symmetrical patterns here that establish the axis of symmetry for these different things, while also establishing that each area here is the body, and maybe this one here is slightly different shade of green is where the head should be, and at the boundary here there should be the arms, right? And maybe with the butterfly it's a bit more clear, right? You can see the region where the wings should be created, right? So it learns to do this spatially structured pre-patterning that then helps the models to self-organize. Questions? All right. Surprisingly, I thought this would have some benefits in terms of parameter scaling. Like if you match the parameters, and there doesn't seem to be that many that much difference. So if you have models with this is like layer 2, layer 3, layer 4, layer 5, layer 1, 2, 3. It's 1 to 5 essentially, and this is the same 1 to 5 on the siren, but with an NCA added on top. There doesn't seem to be much benefit to this. I thought there would, but maybe the main issue here is that before we had the morphogens being a fixed thing, and now we just use it to pre-initialize the initial state and then run with it. So this is a slightly different setup, and I can talk about why this was the case. Now, if you want to use growth, it's a bit trickier, because you need the spatial pattern, because the spatial pattern is just used as initialization and and you run it afterwards, then you cannot just initialize one value of the seed at the beginning. You need something like a patch for there to be some regularity and structure and variation that can help you break symmetry, right? So we would say, well, maybe we can just like a gastroloid would be in biology, where you have this oval shape, and then you have different patternings along the gastroloid, and then you run it. We can do this. And these are examples of how you can do it. You can see, you just pre-initialize this with a pattern. This is the pattern that it learned. And you can see that there's like a lot of repetition and variation of the, it's like a periodic value. And then from there, you just let it run. Because there is no morphogen pattern anymore after the initial condition, you just can, let's say the system consumes the morphogen and takes it into its state, then you can still see, I think it was really cool. You can still see how you get this signaling behavior, right? You can see it. Sorry. You can see it a lot in this one here, where there's like signaling here, bang, and then here you can see the signal propagating from these different points outwards, and the model is coordinating to figure out what they need to do. So I think this is maybe the system that is most realistic in terms of what it does. The other system, I guess, was more interesting from the point of view of studying the computational properties of the morphogen. And this one is more about, can we get it to do something where we have growth? And by the way, these are all invariant, and the substrate is a torus. There's no boundary conditions that it can use to break a symmetry or anything. It's just a torus invariant NCA, and then we create this pre-pattern and we let it run.
Milton,
yes.
What happens if you slice two of those gastroloids up and put them together? Do you get like a chimera?
I don't know. I haven't tried it. It's a good question. I knew I'm laughing because I knew you were going to ask that. But I haven't tried it. Essentially, I've been working. I've been working this week on putting this together to submit to A-Life. So I've been a bit time constrained on working out extra experiments. But yeah, that is a good point. I don't know what would happen. But I should definitely try.
Super cool.
Yeah, so okay, so maybe so a bit of a wrapping up, just right on time. One thing that we can, I think is main takeaway is that you can use fixed morphogenic patterns to scaffold development, and this enables us to break symmetry better. Models develop quicker, and they develop, they reach their target patterns with more fidelity, maybe not more robustness. Maybe there are mechanisms here that are maybe missing to enable this robustness. I have a few ideas on how that could be done in a way that maybe is, or at least in my view, is more realistic, although I'm maybe open to discussing if they are really more realistic. And yeah, you can get better generalization, which from a machine learning point of view, generalization, from a biological point, I think it translates to evolvability by using them. But we can also learn the pre-pattern scaffolds. I do like that they capture, like they learn these symmetries in the repetition of modules. And I think one thing that I would like to explore later is whether we can get better generalization as well. Like if we take a new example that the model wasn't trained on, generate the embedding for it, don't do anything to the parameters of the NCA. Can you just one-shot generate something new? I think that would be the hope. So this is, I guess, sort of like future work, what I've just said. But I think maybe the more interesting angle that I haven't discussed is maybe like these. Sorry, still don't know what happens with the mouse, with the cursor. But anyway, what can we use the system to say something about biology? This is maybe the thing that I've struggled the most with, and the thing that I would like to do more of, which is, if we take these systems, can we now answer questions that are interesting for synthetic biology, for development, that kind of thing? Not just having these models that are very abstract and very cool to look at, but also just doing more, I guess, hard science with it. So yeah, I guess this is it. I'm right on time. So yeah, thank you. And I don't know if you have any extra questions. I think we've guess we've talked a lot. I have time, but I guess up to you.
Thank you, Milton. That was great. Anyone have a question?
Just clarification, maybe I missed this. The pre-pattern scaffolds experiments that you showed, it's the same NCA, right? The parameters are fixed. The only thing that changes is the initial states or the pre-patterns. Did I get that
What do you
right?
mean? You mean here?
The ones at the yeah, I guess so. Yeah.
These ones.
The examples that you showed at the end.
Oh yeah, at the end, yeah.
Yeah.
So the only thing that changes is the embed. Like for each, you just train all of them. You train the system to reproduce all of them at the same time. So we have like an autoencoder that takes the image, generates some embedding for each image, and then that embedding is fed to this thing where it generates the modulations for, we call them usually called modulations here, to change this. And these parameters, the ones in gray, are shared across all elements in the data set, right? So they're meta-trained, I guess.
Okay.
And the only thing that changes is this, right?
And how does the NCA come into play here?
Well, you generate a pre-pattern. You just populate the visible channels, and the NCA just runs afterwards. Just takes these as the hidden channels are set to 0. This is set to whatever was generated here. The NCA runs, right? And this is what I guess my point was with this particular example here is that if the depth is only 2, you cannot generate the target pattern effectively. You still need the development to run afterwards. And you can make this even more coarse. You can just use a layer 1, and this is what we I guess did here, right? This is a layer 1 siren or implicit function or pre-pattern function plus an NCA, and you get similar performance as just using 3 layers of this pre-patterning. And if you can increase both, and you get better reconstruction.
So you would have to train the NCA as well here, right? Even though if it's the same NCA for all the images.
Yeah. I think the hope is more that at test time you can avoid retraining the NCA. You can just modify the thing that you get from the pre-pattern, and the NCA just runs automatically. You essentially have a really robust developmental dynamics that you can just pre-initialize, and it knows what to do.
Yeah, I think one thing that would be really cool to quantify is how much information do you have to feed in the pre-patterns versus the information in the final patterns, right? So the hope would be that the information you feed would be much lower than what you get out. So, and the larger hope, at least for me, it's like, if you can come up with some sort of programming language of sorts where you can just, like, that's a generalization part, right? But with the aid of a sort of a programming languages for patterning using NCAs. So you just program it using logic. You don't have to like train it in some way, but you sort of use logic to program it, and then the compiler will take care of. Yeah, I don't know exactly what I'm talking about, but I'm just thinking of.
I'm wondering what a program means in this context. I think that's mainly what I'm wondering.
Yeah, I guess the general point is that if you are able to get more out than what you put in, then I think that's kind of what biology does, right? It starts with a single egg, with the fertilization event, and then lo and behold, you just get a really complex pattern in a full-blown organism, right? Yeah. So, and a lot of that is orchestrated by the gene program, and some environmental conditions as well, of course. But I guess unit biology, it's not a single universal genetic program. Every species has a different genetic program. So is biology universal in some way? I don't know.
Yes, I guess some of it must be universal, right? The proteins that you express, and the coding, the motifs that you use within gene regulatory networks and within the metabolism of cells I guess what
Yeah.
varies, that part is universal, at
Yeah.
least. Well, I believe it probably there
Yeah.
must be
Yeah.
some variation across species, because obviously we don't. But at the same time, like
Yeah.
DNA is very similar across species, right? So a lot of it must be repetition. You're just varying. I don't know if anyone has done this experiment, but I guess to some degree they have. But if you, how much can you modify, for example, Drosophila? How much can you modify it by just affecting the initial conditions of the egg? I'm sure someone has done this. I imagine it's a non-trivial amount.
Xenobots are a good example of that, right?
Yeah.
You start from a completely different initial state, and then you get something surprising out of it.
Yeah.
Maybe one comment after Santa's question. A program here could be like a construction, like a set of instructions to get to a particular morphotype, right, to a particular shape. So you could ask questions like, when do I need to add what kind of morphogens in order to get to a complex target shape? When you train it on a few base shapes, for instance. I think Alex Mortensov, he mentioned that his life goal will be to have at some point a house that is growing itself, and it's producing all the food and then the medicine he's needing. So that would be kind of a path towards that, right? So like a tiny digital version of it. So what does it take to grow a house or some more complex shape out of these morphogen guided NCAs, for instance?
Well, I guess you could always do, for example, if you have a new target, you can always put the target here at the end, freeze everything here, and do back gradient descent on the embedding space, right?
Yeah, so but I mean during development. It's a generalization experiment. So during development, so how do you need to inject morphogens at a given morphotype snapshot, so to say, in order to get it to update, to change towards a more complex shape, for instance. So if you get to the flowers, for instance, what does it take to elongate one petal, and then from there you radiate out again in order to build some fractal or mandala? Just a thought.
Yeah, I guess that would be a good test. I think the one thing that I wanted to explore at some point was how to, you create a pattern, you run it, you generate something. At what point do you, for example, have a hierarchical system, right? Or that this particular phase of development stops. Now you're in a bigger substrate. Do the whole process again. If you iterate over that, can you just do this fractal thing and just generate something very large? The main issue with this is that once you have too many layers and you have too many iterations of the NCA, it becomes really hard to train, at least with backprop. We can discuss later about like evolution and whatnot, but I think there's someone trying to ask a question, but I don't know. He has a raised hand.
Yeah, Chris.
Yeah, have you or do you know of anyone who's used this kind of technique to actually replicate some of the canonical results from homeotic changes in Drosophila, where the role of morphogens is pretty well understood? So could you train it to make a fly or a butterfly and then get it to duplicate the number of wings or something like that by controlling what morphogens are present when? Is that a plausible experiment with this sort of framework?
I believe so. I would say yes. And this is sort of what I was trying to get at. Or I guess what I was thinking, well, how can we, I guess maybe the limitation is I don't know what the canonical experiments are per se. Like I know that Drosophila people have done like things like, change the homeobox gene expression, and then antennae suddenly changes to a leg or something, But I don't know if that's a canonical experiment. But yeah, I think you should be able to do it. There should be some way to do this, and I would definitely be interested in figuring out how to do it. Like, can we take this experiment, maybe format it in a way that's amenable to these systems? Maybe, yeah, or maybe we need to modify the system in some way. But yeah, like doing this sort of thing and see if we can replicate. I think that would be interesting. I guess maybe the question that I would have is, what would that tell us? Is it just a matter of seeing can we do it, but would it tell us something about biology? Would it help us answer some question?
Well, I think it would be a kind of a benchmark almost, because one knows in some of the cases in Drosophila what's going on. So the question would be, can you replicate a simulated morphogen to make the same thing go on? I mean, the example that I always find so striking is I think it's a double mutant of bithoraxoid and something else that was published in the mid-'80s. One of the early papers on the bithorax complex, where they were able to get the Drosophila haltere to develop as a full wing. So instead of a two-winged fly, you had a four-winged fly. So could you modify, say, the butterfly example in a way that instead of a four-winged butterfly, you got an eight-winged butterfly by altering some anterior posterior morphogen gradient? Yeah, I think it would be an interesting example because so much is known. Anyway, just a question about.
I would be. I mean, if you can send references my way, you know, these were the, these are like the experiments that people have done and how they've, where I can see what. That I think it should be possible. Yes, I think there should be a way to do it.
Okay. Yeah. Thanks.
No, thank you.
I could look for that old paper.
Someone already did something in the 80s for sure.
Yeah.
A similar kind of can this help to explain it problem, though not on something as nearly as well understood as Drosophila, is not sure if you've ever seen. Mike likes to show this really nice picture of a gall wasp, tricking a tree into altering its shape to make it like to make a nice nest for the wasp. If I remember, it's still a tree, but the wasp has tricked it, presumably by who knows. Yeah, there you go. Ben has a link to it.
Let me open this so that I can. Okay, I've saved this for later.
Yeah, I just sent the four-winged fly paper to the chat.
Yeah, I've opened. The galls and the equilibrium from snapshots dynamics, and this one's okay. I have them. Maybe I have to stop sharing because I can't see you all. Yeah, thanks for the references. I'll definitely take a look at these.
I'm not sure anyone knows that the trick involved in Go Wasp is morphogen field changes.
I guess in some sense, like the only way to hijack the developmental system and make it work is by introducing some chemical in the medium, right? And then which changes its gene expression and makes it do something that changes its internal metabolism. So in a way, it can be now. Is it a morphogen gradient, maybe in the same way that this is a morphogen gradient, which is, I guess that's semantics at that point. But any other questions, I guess? Well, then I guess, thank you all for inviting me. Thank you for the references. Thank you for the questions.